And so up next, we have Michael Porter from the Army Corps of Engineers, and his presentation will be Environmental Flow Analysis for Rio Grande Silvery Minnow Recruitment. All right, Nick, you're all set.
Okay, I need to first off thank Rich for queuing up this presentation. I also want to acknowledge Aubrey's work on habitat suitability for reclamation. A lot of the work she did for reclamation contributed a great set of environmental metrics to the process that we're going to discuss today. So moving right along, this presentation is focused on environmental flow, and there's the Brisbane Declaration for defining environmental flow. But specifically, we want to focus on environmental flow management for the silvery minnow. And even more importantly, we want to focus on the flows that inundate the floodplain and create the habitat, as Rich just discussed, for successful reproduction and survival of larvae. So the Rio Grande has been under agriculture for hundreds of years. We've had various bouts of increasing aridity since 1900. Diversion dams constructed 80 plus years ago, and those factors have conspired to extirpate several species that have egg drift as their life history. And what we're interested in is how the silvery minnow, the floodplain inundation, and that connectivity really has helped it persist through the subsequent decades since diversion dams and flood control dams have been installed on the Rio Grande and the Chama. So we're taking what we call a functional analysis approach, and we want to evaluate the relationships of a couple different metrics related to, for the fish metrics, the spring-summer recruitment and the fall population index, and compare them with environmental metrics such as seasonal hydrology, as Rich mentioned, magnitude and duration, and habitat suitability based on hydraulic analyses that Aubrey's done. And what we're trying to get to is understanding what the minimum flow, spring runoff flow, needs to be to produce at least a small population of silvery minnows in a year, and use that to inform water management strategies and possibly restoration design criteria. So this is a hypothetical graph showing the parameters of interest for recruitment flow. We're interested in magnitude, how high that flow is that gets up onto the floodplain. We generally want to see what peak flow looks like. We're interested in duration. And since peak flow tends to be fairly narrow, we're also interested in what we call maximum minimum flow. What is the longest duration that will stay on the floodplain to fulfill silvery minnow life history? And that's kind of what Rich was getting at in some of his slides there. What is functional analysis? For this particular study, we want to evaluate the relationships of fish metrics. In particular, we're going to focus on larval metrics and environmental metrics. And the way we do this is load this data up into a program in R and basically run simple linear models comparing the patterns of the fish metrics with the patterns of the various hydrologic and hydraulic metrics and see what the correlation is. And so pairs of metrics that have a higher correlation in this very simple linear regression, we think may be more important for, or maybe closer to what's important for the silvery minnow. And generally these will have a lower AIC score as well. But we also need to circle back. Just because we have a good metric, good high correlation with a pair of metrics, we also need to be able to circle back around and tease out the ecological and biological relevance of these metric interactions to see if they actually make sense. So, looking at fish metrics, we developed a number of fish metrics. The best performing ones were what we call recruitment slope. You see the formula here in the top table, and it's basically just looking at the increase in the number of larval fish from the standard monitoring that ACER does over a period from May into early August. And then the October index, we put that one in for comparison. We developed a couple other indices. They performed all right. What you see in the second table is that these metrics, the fish metrics, are correlated with one another. And that makes sense because they're derived from the same data set. For comparison, I just plugged in some of the metrics from the spawning monitoring. And what you see is that that data is even noisier than the fish data and has a lower correlation. So our emphasis is really going to focus on recruitment slope and October index. And just to give you an idea, this is a graph by year showing the different indices. The actual, what they represent, what the symbols represent is not particularly important. What we want to show is that there's a significant amount of variability between the indices as you go through the years, but also that there's a reasonable range of variability for each of the indices that we looked at. And that becomes really important because it helps us tease out the relationships when we start looking at some of the environmental metrics. The other thing we did is I'd originally log transformed some metrics and went back through and just took the straight metrics without a log transform. And we actually got better information with the untransformed metrics as we went through the comparisons with the environmental metrics. So very noisy fish data. And what we're trying to do is combine noisy fish data with very noisy hydrology data and see if we can tease out some useful patterns from that. So Albright did a really good job with developing some hydrological metrics, as we see here, and also she translated those into hydraulic metrics and did a much better, more useful job of developing the metrics than I had done. But what it comes down to is these metrics should be biologically important and environmentally relevant to the species and to the ecosystem. Okay, so starting off with the hydrological flow metrics. And these are ranked from higher correlations to lower correlations. On the left side of the graphic here, we have recruitment slope, the R squared and the AIC values. On the right side, we have October index. And what we start to see is that pattern that Rich expounded on, the 14 to 22 day metrics tend to perform a little better than some of the other metrics. I just pulled the top 15 or 16 metrics from each of these series of analyses. We also see that similar metrics occur for the October index. The R squares are not as high. And that makes sense because October is a couple months past when the recruitment metrics were calculated. So it's also factoring in mortality and fish survival, not just the production of offspring. Okay, so here's one of the key features is larval habitat suitability. This is some of the work Aubrey's done for reclamation and showing some of the patterns with surface area as a function of flow increases fairly. This is a log scale on the y-axis. We see it increases, and then taking habitat suitability indices based on the depth and velocity that's relevant for the larvae, we see that those areas are not as high as for the total surface area or the overbank surface area. And when we plug those hydraulic habitat area metrics into the functional analysis, once again, we see some really good metrics at the top end. Keep in mind at the bottom end, because of the high variability, we have metrics that have very low R-squared values. But similar trends, looking at surface area, the overbank velocity criteria. So lots of interesting patterns, but we see some trends that are useful that we can translate into criteria for flow. So taking, just focusing on the 14 and 21 day maximum minimum flow ranges, flows above 2400 CFS for 21 days or shorter. Those flows tended to have really good recruitment slopes and produced better October indices. The threshold flows are intermediate between those, and those flows are between 1100 and 2100 for 14 to 21 days. And that matches up with what Rich was talking about with the mesolarvae swimming ability and the metalarvae swimming ability. So that has some, we think that has some biological relevance for producing fish, as the minimum flow volume to get onto the floodplain, create habitat, and the minimum duration for supporting those larvae to a swimming stage. And then you see at the bottom, the pore flows, that's correlated with low recruitment, low October index. So that's the range we're trying to get above for recruitment. I have lots of people to thank, including Kyle McKay, John Hickey, Nabil Shafiq, Ryan Grunwald, Susan Biddick, and Lynette Geeson, all with Corps of Engineers. Many other folks. We really appreciate the work that ACER's done in producing this data, because it really helps us hone in on some of the factors that are important for the recruitment or habitat. Thank the Rio Grande Endangered Species Collaborative Program, and then some other folks who've contributed ideas along the way. So with that, if there's time, I can answer questions.
All right, Mick, thank you so much. Unfortunately, right now we do not have time for questions, but I just want to remind people, please be sure to ask them though, because we will make sure that they get answered at some point, whether during the break, lunch, or at another time. So